So it looks like a great session tonight. And our first speaker is Sophien Kamoun, who's coming to us from the Sainsbury Laboratory, and he's going to talk about effectors.
All right. Thank you very much. And I also would like to thank the organizers for a great meeting and also for the opportunity to start this session on plant-microbe interactions. And I would like to start by following up on Jeff's wonderful keynote talk. Jeff told you about how plants in nature are associated with all sorts of microbes. I want to re-emphasize that point and emphasize also that it's not just bacteria, but also there's a number of fungal endophytes. In fact, all plants in nature are associated with fungal endophytes. And there's a number also of mutualist parasites that are associated with plants. And really, in nature, microbes interfere and impact all aspects of plant life. And this is, if you're not aware of this work, I really encourage you to go and read about it. It's work by Rusty Rodriguez, Marilyn Rusnick, on how endophytic fungi really, and sometimes their symbiotic viruses can impact plant life. This is from Yellowstone, and the fungi, the fungal virus symbiosis, enables plant to grow at high temperatures, so it has huge impact. One of many examples where these microbes have huge impact on plants, not only parasitic effects. And also we all know that microbes and the science of plant-microbe interactions had a lot of impact on plant biology. There's a number of examples listed here. Olivier Voinet told us this morning about P19, a wonderful protein that's produced by a virus that binds siRNA, and we're all familiar with how the study of plant viruses has impacted our knowledge of RNAi. This is a nice example also. This is work on Gibberella fujikuri, and it's a fungus that causes foolish rice, this really tall rice, and this is due to the secretion of gibberellin. In fact, gibberellin was first discovered from this fungus. And of course, work by Jonathan Jones and many others has revealed the first LRR type receptors in plant thanks to the study of their response to plant pathogens. So why these pathogens do all this, and all these perturbations are easy to understand because a lot of these pathogens are obligate parasites. This is a rust fungus that is totally dependent on its host plant, Bucera stricta, to really go through its cycle. And it has evolved very spectacular mechanisms to interfere with plant life, plant development. So what you see here, this is a normal plant that's uninfected. But when it's infected with the rust fungus, you get these structures, these pseudo-flowers, if you like. It actually mimics flowers in many ways. You have the shape. These are leaves that are organized in flower-like arrangements. There's the yellow color, which is caused by the rust. And there's also the secretion or the production of volatiles that are very attractive to insects, in particular flies. And this enables the rust really to move from one plant to another and go through its reproductive cycle. In fact, there were experiments where the plants were caged and the rust would actually die. So we've done some expression profiling on the pseudoflowers, and there's huge changes. There's over 500 genes that are perturbed by the rust compared to the uninfected plants. There's a shutdown of auxin. There's suppression actually of production of plant volatiles so that the rust can synthesize its own volatiles. So it's one example of really spectacular effect that a pathogen can have on a plant. And this is something we can easily visualize. We can all imagine the molecular mechanism underlying a lot of these interactions. It can be equally spectacular. So how do the pathogens, how do these microbes affect plants? And the best definition of this concept of effectors, so effectors, microbial molecules that perturb plants, is really by Richard Dawkins. And I love this book, The Extended Phenotype, the long reach of the gene. And I like to think of the long reach of these effectors that are, of course, coded by genes sitting in the microbial genome. But really, the phenotype extends to the plant, as in the pseudoflowers. And in the case of the volatiles, they even extend to a vector in this case. And this is really a spectacular concept, one of the most vivid examples of this extended phenotype of Dawkins. So the model, and Jeff introduced this, and I would like to revisit a little bit this concept, because I approach it more from the microbial side, from the pathogen side. So I'd like to start from the microbes. We know that bacteria can inject proteins inside plant cells. Fungi or mycetes also form these structures called hostoria, which also enable them to secrete proteins inside plant cells. And the reason they do that is to interact with plant targets, plant molecules, perturb plant processes, and essentially anything that the effector can do that will improve the ability of the pathogen, the microbe, to colonize the plant will be selected and will end up really evolving as an effector. So maybe one of maybe the most spectacular effector actually that has evolved in a plant pathogen is the TAL effectors, which are produced by bacterial plant pathogens, Xanthomonas, and I think many of you must be familiar with this. These are repeat proteins that bind DNA directly. One repeat binds one base, and there's a code that underlies this. And in fact, now this effector, its reach has gone very far because it's now used in biotechnology and synthetic biology. And I love also the structure of this effector that was published a few months ago, work involving Adam Bogdanov. And this is a spiral, really, that spirals around the DNA. And so I would like to see at Cold Spring Harbor maybe at some point a sculpture really for the TAL effector that wraps around the double helix. It's a fantastic effector. It's a very spectacular one. So the concept, this just summarizes what I've been telling you, but the concepts I'd like you to focus on, the bottom two here. First of all, if you think about it, these effectors, operationally, they are plant proteins. Okay? So I know most of you are plant biologists, but think about this for a moment. Some of the most spectacular plant proteins, some of the most amazing plant proteins are not coded by plant genomes, but are actually coded by pathogen genomes. And the second aspect is that we can really exploit these effectors as molecular probes to understand various aspects of plant immunity and plant biology. And this is something, of course, that historically has been validated more than once. So back to this cartoon and really getting a little deeper, and again, some concepts that Jeff covered the first night is that these effectors, occasionally, they actually activate plant immunity. Okay? So there are plant receptors, usually NBS-LRR receptors that can detect the effectors directly or indirectly. And once an effector is detected, then the plant will win. We get effector triggered immunity and resistance. So these effectors can occasionally, a subset of them on certain plant genotypes will trip the wire and activate immunity. But there's another layer of resistance, and this layer is a surface mediated by surface receptors, usually receptor-like kinases or receptor-like proteins. And this can detect molecules that are in the apoplast. Sometimes these are known as pathogen-associated molecular patterns, or PAMPs. And the activation of the perception of these molecules and their activation, as we heard yesterday from Genshin, activates PAM-triggered immunity, another form of immunity. What's also interesting here is that many effectors, in particular the bacterial effectors, have evolved to suppress PAM-triggered immunity. So it's a very complex interplay with a lot of coevolution going on, with plants fighting back, evolving new receptors, and then the pathogen also evolving the ability to suppress these various pathways. So really to conclude the introduction, we can summarize our field with these 4 major classes of molecules. And I know at Cold Spring Harbor we're not allowed to tweet, but we're allowed to show tweets, I suppose. And I like this tweet I put a while ago that some of my colleagues were complaining that our field is all about effectors and receptors, where at least it's not oxygen and transcription factors. Okay, so now I'll shift gears and tell you more about my own research. And I study the potato blight, the Irish potato famine organism. This is quite an important organism. It's critical to global food security. Potato is the 3rd most consumed crop plant. And it's not a fungus, it's an oomycete. It has a filamentous growth, but it's actually unrelated to fungi. And this pathogen causes disease on solanaceous plants. The crop plants are potato and tomato, but we actually like to study Nicotiana benthamiana. There's a very nice interaction disease interaction between Phytophthora infestans and Nicotiana benthamiana, at least some strains of Phytophthora infestans. And the reason we like benthamiana is because we can do all these transient agroinfiltration assays. Of course, these assays come with caveats, and we can always follow this with transgenic plants and various other assays. But it's really a fantastic system to test and do screens and test number of constructs and really select the most interesting combination and the most interesting phenotypes. So we know already also that in Nicotiana benthamiana, there's some basal resistance to Phytophthora infestans. You can clearly see it here that if we knock down using virus-induced gene silencing, we knock down BAC1 or SERC3, as we call it in solanaceous plants, we have a very dramatic effect on the infection. So we know that wild-type benthamiana exhibits quite an effective basal resistance against Phytophthora infestans. So this is really a useful experimental system for us to play with. Okay, so this is Phytophthora infestans in all its splendor. This is a red fluorescent strain, so you can clearly see the different stages of the disease. We have hyphal tips really pushing into green, healthy tissue of the plant. We have this biotrophic zone. This is where the pathogen is associated with living cells, very closely associated, as I will tell you in a moment. And then there's a necrotrophic phase where the pathogen now kills the plant and actually grows and sporulates on that. During this biotrophic phase is when these hostorial structures are formed. So the hostoria are essentially hyphal tips that push into the plant tissue, still surrounded by a plant membrane, or plant-derived membrane, I should say. And this is thought to be the site of secretion of these effectors. Okay? So this is where the effectors are thought to be secreted. There might be other sites too, but there's some data at least suggesting that that's where the effectors are secreted, where they're interfering with these targets. And indeed, we do find that the effectors display a very distinct pattern of expression. There's a peak that coincides with the biotrophic phase, and that's a very sharp peak. This is a log2 scale expression profiling curves. Okay, so what are these effectors? We have a number of different classes. We have extracellular effectors or apoplastic effectors that don't enter the plant cell. And these are mostly inhibitors, like protease inhibitors. I'll mention about these protease inhibitors in a moment, so keep that in mind. And then we have these different other classes of translocated effectors that we have two major classes in Phytophthora, the XLR effectors and the crinklers. These have signal peptide translocation domain. This region here is quite conserved, defined by these conserved sequences. And these allowed the proteins to translocate inside the plant cell. What's remarkable about these effectors is just the number. So we have 550, for example, RXLR effectors annotated in the genome. We're quite confident about this because of this signature sequence that they have. So we're quite confident about these annotations. That's really a remarkable large number. It's a very plastic family, a lot of diversity. I'm not going to cover that. There's a lot of interesting genomics underlying this. Okay, so the question now is, what do these effectors do? And this is the part I want to focus on for the remainder of the talk. So, of course, some of them will trip the wire and activate immunity, and this is Effector Triggered Immunity. I'm going to talk very briefly about this. I think Brian will tell you more about that. And then, of course, another aspect of the activity is Effector Triggered Suppression of immunity, where many of these effectors are able to suppress immunity. And I'll tell you more about this, and Jonathan, I think, will talk about ETS too. So I'll tell you essentially three short stories about three different effectors in which we made some progress. In all cases, the approach is, as Jeff mentioned, is to find targets of these effectors and really let the effector guide you in understanding or finding something important on the plant side. So this is an effector called AVR2. This effector associates with, in fact, binds kelch protease phosphatase, kelch-type protease, no, kelch-type phosphatase, BSL1, which is, doesn't seem to be involved in brassinolyde synthesis or brassinolyde response in solanaceous plants. But in any case, this effector targets BSL1, and this association between AVR2 and BSL1 is what is detected by immune receptor, by the immune receptor R2. So we know that most likely BSL1 is modified by AVR2, and the modified version is what's detected by the immune receptor. Another effector is AVR3A. This is quite a complex effector because we have many activities for this effector. It's recognized by an immune receptor called R3A. And this is mainly mediated by these two naturally polymorphic residues where some mutants of AVR3 or some natural alleles of AVR3 can evade recognition by R3A. There's another residue that is critical for the suppression of immunity. This is the suppression of the immune response triggered by another secreted protein of Phytophthora, INF1 protein. And this is dependent on the binding of AVR3A to an E3 ligase called CMPG1, which is required for the plant immune response, or the response at least to the INF1 protein. And then there's a third activity of AVR3A, which is binding phospholipids through a pocket here, that's a positively charged pocket. And this is also important for the suppression of immunity, but not the recognition by the immune receptor. So there's a number of different activities for AVR3A. We know that these are dependent on CMPG1, and particularly the suppression of some immune pathways. But there's other alleles and mutants of AVR3A that don't do this. But Angela Chaparro-Garcia, who was at this meeting, discovered that these other alleles of AVR3A can suppress another response, and this is mediated by bacterial flagellin, FLS2. So we suspect that, of course, Phytophthora doesn't produce flagellin, so we suspect that there is a PAMP that uses a similar pathway, and we don't know this PAMP. But the interesting aspect here is that the other alleles of AVR3 have an additional activity, and Angela, and I think many of you were at her poster yesterday, decided to study how these alleles suppress the flagellin elicited response. Again, a couple of slides on this topic. So Angela discovered that there is a target for this effector for AVR3. It's a GTPase. And this particular GTPase is interesting because it localizes at the neck of the hostorium. You can clearly see here, this is again the red fluorescent Phytophthora. There's hostoria formed here. You can also see them there. You can clearly see how the GTPase in infected cells focally accumulate around the hostorium. And then Angela went on and showed that if we silence this GTPase by using RNAi approaches, AVR3A is now enabled to suppress some responses triggered by PAMP and PAMP-like molecules. Okay, so that's the second story. Then the last story I want to mention is really perhaps the most interesting aspect of studying the effectors is to use them as probes to study processes that are difficult to study using, for instance, a genetic approach. And one of these processes is the focal immune response, or the very dramatic reorganization that occurs in plant cells when they are invaded by microbes. And this is known for a variety of microbes, as you can see here. So we're interested, of course, in the okay, and then the question is, are these perturbed by effectors? So we're interested in the hostorial interface. This was always a challenge to study it. So Sebastian Schornack in the lab came up with methods to visualize the hostoria in Nicotiana benthamiana infected plants. And this is even more powerful because he can use agroinfiltrated leaves, so transiently expressing various constructs, and then inoculate them with Phytophthora and then visualize the hostoria. So again, the hostorium, there's a membrane that surrounds the hostorium, and this is probably plant-derived. And you can see here that remorin, which is a plant membrane-associated protein, very nicely wraps around the hostorium. And then the base of the hostorium is labeling with callose, but not the rest of the hostorium. So Sebastian then set up a screen, and he tested a number of effectors, of Phytophthora effectors, expressed them transiently as GFP tags and inoculated with Phytophthora, and looked at whether they relocalize, whether actually tagging along into these focal responses, maybe some of the vesicles that are focally accumulating around the hostoria. And he discovered that this one effector, AVR-BLB2, when it's expressed as a GFP-tagged protein, normally this effector localized to the membrane, but in the infected cells, in the hostoriated cells, dramatically relocalizes to the hostorium. And work by Torga Boskurt, another postdoc in the lab, in collaboration with Joe Wynn and Alex Jones, our proteomics person, revealed that AVR-BLB2 associates with the protease, C14, this is the protease, and this particular association we're not sure if this is direct or not, but this is certainly part of a complex that AVR-BLB2 pulls down in the plant. This was interesting to us because at the same time, our collaborator Reinier van der Hoorn discovered that the same protease, C14, was inhibited by these apoplastic effectors. So these effectors that are present in the extracellular space. So it seems like this protease is targeted by multiple effectors of Phytophthora at different sites. And we also discovered that this particular protease, if it's labeled, expressed as GFP protein, also accumulates, focally accumulates around the host area in a very dramatic fashion. So Toga went on and confirmed the association. Again, we don't know if this is direct or indirect, but he discovered what AVR-BLB2 does to the protease. He found that AVR-BLB2 prevents the secretion of this protease into the apoplast. So this is the model. We think as the hyphae is growing, the plant is secreting this protease. There are apoplastic effectors that are inhibiting very strong, very potent inhibitors of this protease. And this is probably happening at the hyphal tip, maybe other parts of the hyphae. And then as the hyphae grows, there's hostorius form. The protease is focally accumulating now around the hostorium. And then now Phytophthora has a different effector, AVR-BLB2, which is preventing the secretion of this protease. So this is really opening new avenues for us because it forces us to start thinking about this interaction both in temporal and spatial fashions. And I think this is really exciting topic for the future to really think about this interaction in a more dynamic way. This is also interesting because the extrahostorial membrane, so this is the plant-derived membrane that surrounds the hostorium, we know has a very unusual composition. So for example, this is now a labeling of the Phytophthora hostorium with synaptotagmin. So this is membrane-associated protein also that you can see very nicely accumulates around the hostoria, as well as in the membrane. And then if we take PAMP receptors, so pattern recognition receptors, some of the surface receptors involved in immunity, like EFR, in this case, even though this receptor is nicely labeling the membrane, it is completely lacking from this extrahostorial membrane. So the question for us in the coming years would be to understand this process, these differences. Is the pathogen secreting effectors that are specifically targeting these various proteins, various processes or not, and how does this function, etc. And this is, we have a number of candidates study this because there's other effectors that accumulate around the hostoria. So you can see here one particular effector that accumulates in vesicles that seem to be more abundant at the base of the hostorium. We have another effector there at the base of the hostorium, one at the tip. So again, these are really fantastic molecular probes to dissect this process that has been really intractable by other means, especially genetics. So I'll finish there, and I think I mentioned the people involved in these various projects. AVR2 was Diane Saunders' work. AVR3 is Angela in collaboration with Toga, and then the AVR-BLB2 part was Toga and Sebastian working as a team, and I would like to thank my colleagues at the ScienceB Lab. So I'll stop there. Thank you very much.
Sophien?
Yes, you said in the beginning that these translocated effectors, that they have a rather conserved domain in order that is necessary for the translocation.
Right. Yes.
And I was just wondering, is that a good idea actually for an effector to have a conserved domain?
Right.
And shouldn't the plant use that actually as a PAMP?
Right. That's a very good question. So we don't actually know what happens to that domain following translocation. It could very well be that it's cleaved. We don't have evidence for that, but that could be happening. But that's a good question. And but I should say that that domain is actually quite diverse. It's not. I mean, there's a signature there that our XLR sequence, which is quite conserved, but it is relatively diverse. But that's a good question. And as far as I know, nobody has identified the plant receptor that detects that domain. Most monocots are resistant to Phytophthora, so maybe that's exactly. Maybe that's the explanation.
Sophien,
yes, Jeff.
So, Miko, type 3 secretion needles can act as PAMPs in some systems. So that directs your question. Yeah. I'd like to go back to BSL1,
yeah. Right.
Which seems to be a target for an effector that's also associated with an NBS-LRR receptor. Do you know Yet whether phosphatase activity is required or altered
Right.
or activated by association with the effector, and whether that's
Right.
the mechanism of activation.
No, we
of
don't
the
know that
NBS-LRR.
yet. We haven't been able to make mutants that, catalytic mutants, if you like. Yeah. So we don't know the answer yet.
So, given the fact that the Phytophthora has so many RX LR effectors, and compared to especially Xanthomonas ringi and Arabidopsis system, for
Right.
example, and also the infection process is so much complicated with the formation of Hosteria. Is that possible? There are infection stage specific effector proteins.
Yes, so I didn't address the question of their expression. So the first thing, even though there's 550 RxLR effector genes in the Phytophthora infestans genome, in a given strain, we only have about 120 that are expressed. So there's huge expression polymorphism going on between different B infestans strains. So that's an aspect. I mean, there's redundancy built in the system, and I mean, this is another topic. I'd give a whole talk about that. But I mean, the genome size, the plasticity is, there's plasticity at all levels, including at the expression level. I mean, the question of the different phases, I mean, yes, there is actually different waves. There are effectors that are expressed early in the interaction and don't peak later as some of the others. The ones with an avirulence activity, they all seem to peak in the same way. And I think the ones that are expressed too early, and this actually recent paper from our collaborators in the Netherlands, suggests that those don't actually activate a strong enough immune response to block the pathogen. So there seem to be a subset that are expressed at a particular stage, and those are the ones that tend to be effective targets, essentially, for the plant to go after with their immune receptors.
Okay, so thank you, Sophien.
Sure.